The mammalian immune response can be divided into two branches: innate immunity and adaptive immunity. Innate immunity is an intermediate non-specific response that cannot perfectly distinguish different pathogens. Furthermore, once the innate immunity response is depleted, there is no memory left of the encounter with the pathogen. This is when the adaptive immunity becomes crucial. Adaptive immunity recognizes specific components of specific pathogens and retains the information of the encounter. This is due to the presence of billions of cells circulating. Lymphocytes of adaptive immunity, often simply referred to as lymphocytes, being far more numerous and more important than those of innate immunity. So what do lymphocytes recognize? Lymphocytes recognize any molecule that may belong to a specific pathogen, for example, a protein of a bacterium, a saccharide component of its cell wall. The macromolecule recognized by the lymphocyte is called antigen, as it can generate a response against itself. Each lymphocyte is able to recognize only one specific antigen. Called naive lymphocytes, non-reactive lymphocytes circulate in the bloodstream until they encounter the antigen and become reactive, thereby becoming effector lymphocytes. Once effector lymphocytes, their function differs depending on the type of the microenvironment. In general, lymphocytes can be divided into two groups, differing both in their antigen recognition mode and the type of response they initiate. These are the B lymphocytes and the T lymphocytes. B lymphocytes recognize the antigen by means of the BCR, B cell receptor, an immunoglobulin-like molecule embedded in their membrane. The BCR is a macromolecule which, like immunoglobulins, is often depicted as Y-shaped for educational purposes. It consists of 4 protein chains, 2 of which are bulkier, heavy chains, while the other 2 are smaller, light chains. Taken together, the 4 chains comprise a constant region, the root and proximal part of the Y branches, and a variable region, the end of the 2 Y branches. The variable regions have the antigen binding site at their end. Therefore, each antibody has 2 antigen binding sites, one for each variable region. The constant region of the BCR, on the other hand, faces the cytoplasm and activates the lymphocyte signal transduction after the antigen binding. Once the B lymphocyte becomes the effector, it turns into a plasma cell. Plasma cells are responsible for producing a large amount of soluble immunoglobulins that recognize the same antigen recognized by the BCR. These soluble immunoglobulins have a specific constant region for binding with a specific cell type. For constant regions, there is in fact a certain variability, which is much less extensive than the variable regions and which determines antibody binding to a given cell type rather than another. In any case, each lymphocyte produces only one type of immunoglobulin and only one type of constant region and a variable region that is only able to recognize a specific antigen. This means that a single clonotype of antibodies is produced. At this point, we need to clarify one thing. The variable portion of immunoglobulins is not capable of binding all portions of an antigen, but only binds a portion that is called an epitope, also known as antigenic determinant. It goes without saying that several antibodies can bind different epitopes on the same antigen. This is very important because it allows a pathogen to be recognized from different points of view and facilitates its removal. Let's take a look at T lymphocytes now. Like B lymphocytes, they are provided with an antigen recognizing receptor, the TCR, a T cell receptor, which is a molecule similar to the BCR, although smaller in size. Unlike the BCR, the TCR is not able to recognize a specific antigen directly. To perform this function, the TCR needs the antigen to become attached to some protein macromolecules called major histocompatibility complexes, MHC. There are various MHCs, although the most important are the MHC class 1 and class 2, which have different functions and activate different subtypes of T lymphocytes. MHC and antigenic peptide complexes are typically expressed by antigen-presenting cells, APCs. Dendritic cells are by far the most important APC. However, under certain conditions, macrophages and B lymphocytes themselves can also act as APCs. Once the MHC peptide complex has been recognized, T lymphocytes become effectors and can perform 3 different functions which cannot be concurred in the same lymphocyte. 1. Cytotoxic function. T lymphocytes come into contact with the cells that expose the antigen they have been activated for and then kill them. 2. Helper function. T lymphocytes produce molecules that can coordinate the immune response. 3. Regulatory function. T lymphocytes express excessive immune activation and protect the body from the excessive inflammatory reaction. A common function of both B lymphocytes and T lymphocytes is that of the rise of the memory cells, which can remain in the body for a long time and reactivate when they encounter the same pathogen again. What is the potential of lymphocytes in comparison to cells of innate immunity? In other words, why is there need for adaptive cells? All the receptors of innate immunity together can recognize nearly 100 different proteins. On the other hand, if we analyze the lymphocytes of a single individual, it is estimated that they can recognize 108 different peptides, i.e. 100 million. Basically, lymphocytes help us to have a ready and effective response to a range of antigens that the cells of innate immunity alone could not cover. Therefore, we speak of a lymphocyte receptor repertoire, and its complexity is due to the particular gene rearrangements that lymphocytes undergo during their maturation. This process is extremely delicate as it inevitably leads to the formation of self-reactive T cells, which can identify as harmful as those antigens normally expressed on human cells, self-antigens. However, these cells are promptly eliminated during lymphocyte maturation in a process known as clonal deletion. Alterations in clonal deletion mechanisms at various levels underlie some autoimmune diseases. On the contrary, when mature naive lymphocytes are put back into the system, they recognize their peptide. They can proliferate and form many lymphocytes with the same specificity as the original lymphocyte. This is called clonal expansion or selection. We will now briefly look at where lymphocytes originate and the tissues of the body their maturation and activation take place in. Like all blood cells, lymphocytes originate in the bone marrow. However, there is an important difference between B lymphocytes and T lymphocytes. B lymphocytes complete their maturation in the bone marrow, whereas the progenitors of the T lymphocytes need to complete their maturation in the thymus, an organ which is located in the front of the heart. For this reason, bone marrow and thymus are referred to as primary lymphoid organs. Once they are mature, naive lymphocytes enter the bloodstream and begin to populate the secondary lymphoid organs, where they encounter their own antigen if present. These organs are the lymph nodes, the spleen, and the mucosa-associated lymphoid tissues. Although different in structure, secondary lymphoid organs essentially have 2 functions. 1. To facilitate the encounter between immune cells and antigens, which would otherwise be more complex and less likely to take place in the bloodstream. 2 to provide lymphocytes that have not immediately encountered their antigen with survival signals. In this way, they can go back to circulating in the bloodstream, re-enter the secondary lymphoid organs, and eventually encounter the antigen. Secondary lymphoid structures are then extremely dynamic and can swell or shrink depending on the antigen charge at any given time. Have you ever had a swollen lymphoid node after a surgical procedure on your teeth? At the moment, the surgical procedure causes the release of a large number of antigens and inflammatory stimuli, which results in the swelling of the lymph nodes in the immediate surroundings. Lymph nodes are small, somewhat spherical structures spread throughout the body, which form proper chains at the branch points of blood vessels. From a physiological point of view, their role is to collect lymph, an extracellular fluid that forms continuously in the tissues of the body and contains important immune mediators, as well as possible dendritic cells activated in peripheral tissues by contact with inflammatory molecules. The lymph reaches the lymph nodes by means of afferent lymph vessels, which opens the cortical sinus of the lymph node, i.e. in a peripheral cavity of the lymph node. The lymph then passes through a number of areas. The cortical area, which essentially contains B lymphocytes organized in follicles. These may be spread, or if stimulated to proliferate by contact with the antigen, they may create rounded germinal center or secondary lymphoid follicle within the area itself. It should be noted that B lymphocytes are rarely able to activate autonomously after making contact with the antigen, as they require interaction with T lymphocytes and their help to do so. The paracortical area, which contains mainly T lymphocytes. This is a crucial area, as it is where T lymphocytes, activated dendritic cells and macrophages come into contact with each other, as they are attracted by the same chemokines and can create useful interactions for T lymphocyte activation. The dynamics of lymphocytes in the lymph node depend on their activation status. In fact, driven by different chemokines, activated T lymphocytes can migrate to the boundary between cortical and paracortical areas and act as helpers in the activation of B lymphocytes. Medullary area, where there are medullary cords, which are clusters of cells consisting mainly of macrophages and plasma cells. Medullary sinus, which collects the lymph coming from the lymph node. Efferent lymphatic vessels, from which lymphocytes and the lymph come out to reach the systemic circulation. These processes usually take 4 to 6 days. Naive lymphocytes that haven't recognized their antigen also leave the lymph node and recirculate in the blood until they encounter their antigen or die. This is because there is a fundamental rule for lymphocytes, except for memory cells. Lymphocytes that do not recognize antigens for a certain period of time stop receiving survival stimuli and undergo cell death. This mechanism is essential to avoid an overpopulation of lymphocytes in the blood and the lymphatic organs. The spleen is a secondary lymphoid organ located behind the stomach. It has no connection with the lymphatic system, but it is responsible for monitoring the presence of pathogens directly in the bloodstream. It is therefore important to fight all those pathogens that manage to enter the bloodstream. The spleen consists of 2 areas. The red pulp, which is responsible for hemocataresis. This represents most of the organ. The white pulp, which is located inside the red pulp and has immune functions. The white pulp is a cluster of immune cells which forms around the central arterioles, i.e. blood vessels branching off from the trabecular artery to the spleen. The central arterioles are characterized by large discontinuities in the endothelium that allows cells and antigens to rapidly pass through. The white pulp is made up of various components. The periarteriolar lymphoid sheath, PALS, a lymphoid lining that wraps around the whole central arteriole. The PALS mainly contains T cells. It is functionally similar to the paracortial area of the lymph node, since activated T lymphocytes and dendritic cells can meet here. Lymphatic follicles, which are essentially populated by B lymphocytes, like the lymph node. Germinal centers, secondary follicles, surrounded by a crown, may be created here as a result of the activation of B lymphocytes. The marginal zone, which surrounds the crown of follicles, this is populated by the B lymphocytes in the marginal zone, which are innate lymphocells. Unlike the B lymphocytes, these lymphocytes rapidly produce low affinity antibodies to bacterial polysaccharides. Since this production is not antigen specific, they are referred to as lymphocytes of innate immunity. As the marginal zone is the closest to the perifollicular zone, it is where dendritic cells are activated before migrating to the T areas. Lastly, there is the perifollicular zone, which is located between the red pulp and the white pulp. What pathway does the blood follow in the spleen then? From the central arteriole, the branches arrive in the perifollicular zone, which is actually the first entry point for cells and antigens. From here, they can come into contact with the white pulp and trigger the activation of the immune system, similarly the lymph node. The blood is then collected in trabecular veins and flows out of the spleen. Mucosa-associated lymphoid tissue, MALT, are located in the walls of the main hollow organs of the body. This should be no surprise. Hollow organs are in fact precisely the first phase of the pathogens and antigens that we take in from food or air. Moreover, many mucous membranes are populated by the functional bacteria flora, the microbiota, which stays in our body without being affected or destroyed. Think of this, MALTs are so important that they contain as many lymphocytes as the entire organism. MALTs include NALT, nasal mucosa-associated lymphoid tissue, BALT, bronchial mucosa-associated lymphoid tissue, and GALT, gastrointestinal tract-associated lymphoid tissue, which is by far the most complex and extensively studied component, and includes the tonsils, adenoids, appendix, and intestinal Peyer's patches. The Peyer's patches are lymphoid thickenings located in the walls of the small intestine and preside over the control of antigen entry from the gut. From the lumen towards the wall there are M cells microfold, which are the functional equivalent of the afferent lymphatic vessels of the lymph node. These are columnar cells that pick up antigens and pathogens from the lumen. Subepithelial dome, an area right below the M cells that is rich in dendritic cells. This is where antigen loading onto APCs can take place. T cell areas, which form the bulk of the plaque. B cell follicles with germinative centers, which are scattered into the C cell areas and are ready to interact with helper T cells in order to be activated. Efferent lymph vessels, out of which activated lymphocytes can leak to reach other tissues, usually other distant mucous membranes. Experiments on mice have shown that there are only a few hundred antibodies specific for a given antigen in the body, which certainly can't stimulate a response against a pathogen all by themselves. Therefore, when a lymphocyte becomes activated, it stops migrating and proliferates even before becoming effector. In order to do so, it becomes a lymphoblast, that is a very voluminous lymphocyte with an intense nuclear and cytoplasmic volume and large amounts of mRNA and proteins. Be aware that the term lymphoblast is often also used to refer to one of the intermediates of lymphocyte maturation in the bone marrow. Lymphoblasts duplicate at a rate of 2 to 4 cycles every 24 hours for 3 to 5 days. This leads to a creation of 1,000 daughter cells per lymphocytes with the same antigenic specificity, which multiplied by a few hundred different lymphocytes mentioned earlier make up a true action team. Only at this point do the lymphocytes transform into effectors and their fates diverge. The B cells become plasma cells. Most migrate to the bone marrow, from where they secrete antibodies directly into the blood, while some may remain in the peripheral lymphoid organs. T cells, on the other hand, can follow the various fates mentioned earlier. Some B cells and T cells create the immunological memory. It ensures that the antibodies produced upon the second contact with the antigen have a higher affinity maturation process, binding the pathogen more effectively in order to remove it. By the same process, vaccinations succeed in preventing the development of infectious diseases. It is precisely by virtue of the more impressive response set in motion upon second contact with the antigen that many vaccinations require 2 or more doses to refine and enhance the quality and strength of the immune response that is put in place. To conclude this video, let's now try to get an overview of the adaptive response activation, while keeping one essential aspect in mind. The adaptive response needs the innate responses to activate and persist. Thinking of these two responses as two separate things is useful for teaching purposes, but could become misleading in real life. It's important to point out another key concept of adaptive immunity. The encounter between APCs and lymphocytes does not necessarily lead to lymphocyte activation. To make this happen, you need a series of inflammatory signals to support lymphocyte activation. Bearing these things in mind, the inflammation resulting from an infection leads to the activation of the dendritic cells located in the affected tissue, among other cells. This leads to a series of changes in their behavior as they begin to phagocytize the given pathogen more efficiently to stimulate micropinocytosis. For example, the uptake of extracellular fluid potentially containing parts of the pathogen, and to express MHC containing peptides of the pathogen together with the co-stimulatory molecules which are essential for activating lymphocytes. In addition, following the chemokine gradient, they begin to migrate towards secondary lymphoid organs where they will encounter naive lymphocytes. At this level, the naive lymphocytes that continuously recirculate can recognize their own antigen, and thanks to inflammation-induced co-stimulatory molecules, they become activated, proliferate, and become effectors. These will tackle and resolve the infection. Moreover, a part of them will act as immune memory in case of future encounters with the same pathogen.
Thank you for watching. This video was created by School of Biomedical Sciences, Agora. We hope you enjoyed it. If you're curious or have any doubt or question, please feel free to leave a comment below. If you want to find out more about us or want to support our project, click on the following link to visit our website.